how to solve this particular problem. And one important caveat, because we were towards the end of the class, I didn't mention, and I think Nirav came towards the end and asked this question, is okay, I think that has to do with my port. Sorry. See. Okay. So the simplification that we had done actually for that problem, which was implicitly mentioned, is, otherwise at this point you will not be able to solve this particular problem, is that these sites are independent. Otherwise it will be very hard to solve this problem. Okay. So what we had assumed, that if I get a block here of restriction sites, I'll just call this R, we're assuming that this does not impact if I get a block here at the immediate next position and so on, right? So the sequence that we got was GAADTC and we are making this assumption that if I see something like this, it does not actually change what I'm going to see next, which we know is actually not true, because if I shift this by a base, there is some interdependency here, right, in terms of bases. So for example, if I changed this restriction site to be this, will your expected number of, will your variance, actually the expected number will not change. Will your variance of the fragment sizes that you're going to get, is it going to be smaller or is it going to be same? What, why, and why not? So the expectation does not change, because you recall this is always going to be this. However, for this to be true, which is a variance of sigma of xi is equal to sigma of variance, sigma I of variance of xi, there is a special condition required, which is xii have to be IID, have to be independent. Otherwise, this condition does not hold true. We have not discussed the clause at all where there is an interdependence built in, but very broadly, what will happen is there will be a negative factor involved, which is of covariance of xi and xj. This we'll discuss later in the class when we start discussing about covariance. But broadly, the idea is we are assuming if I see a particular motif here, this is called a motif, a sequence motif, then the chance, it does not affect the chance that I'm going to observe it here or not. Right? They are assumed to be IID in all possible sense, and this will be specified. Even if it is not specified, just you can assume, you can just do the best case scenario. I did not expect you to actually go that route where there is dependence involved here. But does everyone see why there will be a dependence? Because there is some dependence here, and depending on how things come about to be, your chance of observing one sequence will indeed affect the other sequence, right? If you see one motif, the other one will not occur, and so on. However, if you had gotten lucky, if there was a T here, then you could do, or let me make it even simpler. If it was this, you would have a lot more interdependence already built in, right? You could keep seeing restriction sites after each other. Okay, if that question is clear, we are now going to solve a slightly different question, which is, what is the probability of observing 2 or fewer restriction sites. You don't have to assume the same, you can just assume that the probability of seeing that motif is P dash. What is the probability of observing 2 or fewer restriction sites in a DNA of 10,000 base pairs? How will you solve this? CDF, okay. How will we do the CDF? Okay, so the question is, so whenever you see a question like this, it makes a lot of sense to write this down in form of a mathematical operation. So just to revise, we had talked about indicator variable where I'll just call it restriction site RS to save time. This is set to 1 if my ith position is the start of a restriction site, and if it is set to 0, and if I have to count the restriction size, then it is simply going to be sigma xi, which I'm going to call this. This is the number of restriction sites, right? I'm asking what is the probability of observing less than equal to 2, right? So I'm just changing that particular notation into mathematical equation. Okay, nothing very difficult here. Is this clear? Okay, how do we solve this? Py equal to 0. How? 5%. Why that is lambda? That is the average number. That is expected number of. Okay, so very good. So that is that's exactly why this problem was invented, right? This is an invented problem to motivate that if I have to do this kind of in this setting, you can always do. So what is this? Like it's nothing actually, right? It's n choose 0. Simply a probability problem, right? You must have either seen this problem. It's simply asking what is the probability of if you toss a coin n times, what is the probability of seeing at least or 0, 1 or 2 heads, less than equal to 2 heads. It's very, very simple in that fashion. Okay. However, there's one slightly easier way to solve this. Often in cases, here we are lucky that we can calculate NC0, NC1 and NC2. But this binomial is maybe, if I gave you 10,000 base pairs, and if I change the question to ask what is the probability that the number of restriction sites will be less than equal to 500. Okay. This will remain the same, but I'll have to continue summing this up to n choose 500, p to the power 500 and so on, right? And whatever, n minus 500. I'm not writing n equal to 10,000, but 10,000 is equal to n. Do you see the problem here? Like this is a computationally hard problem because calculating binomials is not easy. Manasvi, question? Sure. Doubt? Clear? What is it, right? So you mean this probability, right? So I'm exchanging p dash to be p. Basically, what is it? It's the probability of seeing this particular motif, which in this case was GAATTC. So what is the probability of seeing this? Is your p in a sequence of. Okay, good question. So imagine your ACTGs, it's a box where you have a lot of ACTGs, right? What is the probability that if you're drawing 6 draws, you will get exactly this sequence? That's what the intuition of P is. In 3 into 10 to the power 3 base pair sequences, which are of this length, what is the probability that at a particular stretch, you will see GAATTC? Why do I care about it, right? That is because you have an endonuclease, which is an enzyme. So there is a scissor, right? You cannot draw scissors, but yeah, this is scissors. It's going to come and start to make a cut here. And that's why we are interested only in that particular site. Okay. Yeah.
Then the chances of getting GAATTC is more than, let's say, if it is 10,000 base. So isn't probability changing?
Good question. It's a very good question because that's the difference between probability and expectation, right? So when I take a particular sequence, at this point, this motif has been decided because this is what the endonuclease recognizes. Right? So the first question that I'm asking is, if I take what is for any 6 base pair sequence, what is the probability of observing something like this, right? So that is just determined based on what is PG multiplied by PA multiplied by PA multiplied by PT multiplied by PT multiplied by PC, right? This at this point is simply a question of probability. What is the probability that I'll observe this? Okay. The point of expectation is, on average, the moment I see something like this, this will give rise to 2 fragments, right? I'm asking, because of this, on an average, if I have a length of 10,000 base pairs, how many fragments will I see? Okay. For this event has to occur, and it will occur multiple times by chance, because the probability is high or it's small. That will give rise to the number of fragments that you're going to see. So there's a difference between the expected number of fragments and the probability, right? The probability is what is the chance that I will see it in a given motif of length 6, right? Because it could also happen that I get GAAAAC. It won't cut here, but this also has a probability, right? This also has a probability, but this is not going to give you rise to your expectation because it does not meet your requirements. Okay? Every 6 base pair will have a probability of being observed, but it does not give rise to the fragments, right? It is possible that if this probability, almost tends to 0, I will not see a cut at all. So my expected number of fragments should be 1. And that's what we are essentially trying to capture. Okay.
What is N there?
N is the length of the sequence, which is the genome.
Then, let's say for Y equal to 0,
yeah.
You shouldn't take the entire thing, right? The last fiber will never.
Good question. That came up yesterday also. Yeah, that's correct. Yeah, so I won't go from y equal to 1, so this is a good question. If 1 is the start of the sequence and n is the end of the sequence, I will only go to n minus 5 positions.
And then for y equal to 2 also, we can't choose consecutive. Like NC2.
Yeah.
Probability of y equal to 2, the last.
NC2, yeah, this one, yeah.
So with this we could choose like this pair and then the next one also. But then 2 consecutive bases can't.
Good question, right? So your question is, if I have, this is my sequence. In the first, I'm not seeing anything. In the second one, I'm seeing one hit. Now this one hit can occur anywhere actually. So far, I've not told you which position it is occurring. But it is obvious that if it is occurring here, it will not, the other stuff cannot occur here. So in this case, you can ignore what's called the edge effects. These are like edge effects. The reason we are ignoring this is because, this is a very good question which I did not emphasize earlier, is because this is a length pair of 6 and we are working in at the order of 10,000 base pairs. So we're going to assume that these kind of cases, which are called the edge effects, are in a sense ignored. Okay, despite that, it won't change the calculation of your expectation, right? Because expectation does not feature in, it does not take into account this interdependence, because each of this is maybe not a dependent, not an independent variable, but the summation of this, the way you take expectation is going to be the same. What it will change, what does it actually change, is the length of the fragments, or the variance of the fragments that you're going to get. That is going to be different, right? We will not talk about it right now, but it will come in a later part of the course. Okay, so we are ignoring edge effects. Yeah. Sakit, yeah.
Does this mean if we are asked probability of seeing two restriction sites, one restriction site is already observed?
Oh, that's a good question. Yeah, so that will change. That's a totally different problem. But yeah, think about it. Maybe it will be a question in the exam. Yeah, it's two separate scenarios, right? So if, for example, if you've saying that, if you're conditioning on the fact that this is fixed length, maybe a length L, and you're observing one restriction sites here, what is the probability that you'll see an expected, you will see a restriction site here based on some other properties that I will give you. You can calculate that. Yeah, okay. That will become a totally different question. Okay, but you see the problem here, right? If this number, yeah, that won't be IID, right? So in that case, we'll change. If you have a particular sequence, so if I shrink this and if I work in a, so we will do all of this actually, right? Like we will do this case where we have 100 base pair sequence, and I give you a particular site, and I tell you that there is a particular base composition. What is the probability of seeing something as extreme as this in this particular context? We'll do this in few lectures later. Yeah. Yeah.
Then also we can't break this problem into L minus 6 में एक restriction site.
We cannot break down
That basing problem into L minus 6 length में एक restriction site also. Because we have some X length to the left of the first restriction site and some X length.
Okay, all of this will come clear. So all of these are good questions. This will all come up once we start talking about the read structure and the how much amount of, so keep your thoughts very tight. We'll talk about when we start sequencing this. When we take a genome and when we actually start sequencing, these are exactly the problems that we'll worry about. Okay, so just to, if you want to read ahead, you can read about genome sequencing coverage. How is that defined and how is that calculated? These are exactly the same question. This is called, I'll just give you the name, you can look up. This is called Smith-Waterman, Landau-Waterman gap statistics. Okay, so these are sort of equations which were calculated when the human genome was being sequenced. That how much amount of sequencing do you need to do to ensure that you cover the entire human genome, which is 3 billion base pairs. We'll start to talk about it, I think 3 lectures down or 2 lectures down, depending on how things go. Okay, so you can see this is complicated. If I have to do a binomial sum, binomials are anyway very hard or much harder to calculate. So imagine doing 10,000 factorial divided by 10,000 minus 500 factorial multiplied by 500 factorial. It's a very hard number to calculate. Any other ideas you can use here? So he said this already. He assumed that is going to be Poisson, right? Why Poisson? What is Poisson here? Recall the definition of why we had defined Poisson. So we were working with Bernoulli. We summed it up. It became a binomial. And then you're saying it's Poisson. What is Poisson here? Right. Okay. So n is very large, probability is very small. So we had seen a version of this where Poisson is a good model for modeling counts. The counts here being the number of restriction sites when it's a rare event. That rareness is coming because the probability of finding this is rare, right? This is usually going to be very, very small, and your entire sequence, which is n, is going to be very, very large. Okay. So this problem then boils down to a binomial, a Poisson approximation. It's not very hard to derive it actually also. We can approximate this to be a Poisson random variable. Okay, so just to recall, a binomial is represented as n factorial by j factorial n minus j factorial p to the power j 1 minus p to the power n minus j, and I'm saying that you can represent this as a Poisson with lambda is equal to np. Okay, we'll see how this is derived. Any ideas of how we go from there to there? What is the PDF of a Poisson? How does it look like? I always struggle with this. So what is p x equal to x for a Poisson? It's a discrete random variable. This? Okay. Looks very distant, no? Like a factorial and an exponential. How do we go there? Any ideas? And we are saying that this holds true if n is extremely large, p is very, very small. Okay, so let's see if we can do something here. So this is n factorial, right? I can write this as n minus j factorial into okay till there's a mistake. N minus j, this has to go all the way till n, right? So n minus j plus 1, or okay, we'll struggle again. Okay. 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 N minus j plus 1 and all the way till n minus j minus j. Okay. So these two terms get cancelled. I get n minus j plus 1 till n divided by j factorial p j 1 minus p to the power j and 1 minus p to the power n. Okay, what do I do next? Okay. Okay, can we do something here? So if this goes to 1, it's an early premature approximation, right? I think we should do something else. What is that something else that needs to be done? So we have to bring it down to this particular format. What is e to the power minus lambda? Do you see any format here which is e to the power minus lambda? Okay, 1 minus p. Okay. Okay, so this becomes e to the power minus np, or e to the power, this is Okay. Is that correct? And what about the rest? N minus 1 is approximately equal to, but there are so many n's, right? So okay, so if all of them, essentially you're saying that I'm going from 1 to j, right? So this is n minus j plus 1 into n minus j plus 2 up to n, which is j plus j. This is 1 to j. Okay. We have p to the power j. That's right. Okay. So we can multiply each of them here, which is, we can club it to each of them. Okay. And what does this become then? Okay. So this is n times n minus j plus, sorry. So if I write np as lambda, right? If I write my np as lambda, this is lambda to the power j, sorry, divided by j factorial. Okay, does that make sense intuitively? Okay, so what we did essentially here is, this is our j factorial, this is p to the power j, this is a small value which we are multiplying by otherwise a large number. We have assumed all of them to be almost equivalent to np because these numbers are going to be very, very small. Your overall term here will, when you multiply this by np, it's mostly np that's going to add up because everything else is going to be even much smaller. Okay, so if it's also there on the slide, so you don't have to actually do it, but you can set up your approximation such that it looks like it's very similar, except in this case I substituted p to be lambda by n. Otherwise, all we did there is very, very similar overall. Okay. What we are essentially assuming is, or invoking, is this particular property, which is limit of n tending to infinity 1 minus x by n is e to the power minus x, which is what we invoked in here. Okay. Any questions? So that's how a Poisson becomes, that's how a binomial becomes a Poisson. And the reason this is often easier is, now we can invoke this particular number because this is easier to calculate. All the calculations that we're going to do here, for example, even for x equal to 50, this is an easier number to calculate. This is a fixed quantity. This is easier because you can do log of this. So multiplying x log lambda. Similarly, the factorial you have to calculate, but that's not as hard as calculating a very large factorial twice. So how will we solve this particular problem? Yeah, so in this particular formula, where lambda is equal to np, you just sum up x equal to 0, 1 and 2. And that gives you very similar results as a binomial would give you, except that now you're invoking a Poisson version of it. Okay. Okay, so you get 0.57 irrespective of however you do it. Right. So what this actually means, right? Just look at the number. We started from a 6 base pair sequence, and the probability that we get 2 or lesser sites is 0.57, which is almost half, right? So just a 10 kb sequence, which is pretty long, it will be cut at least 2 times, with a very high probability overall. So while we can calculate probability, you should always go back and see whether it makes sense. Even with a smaller motif, which is 0.00, which had a 1 to the power, you know, orders of -6 probability, you're able to actually cut it twice very easily, you know, with greater than 50% chance. Okay, any questions about probability and statistics? So that is that winds up our probability and statistics. You have studied how to calculate, how to sort of think about distributions, how to calculate expectations, how to calculate variance in the IID case. And with that, we have sort of calculated how to look at, how to model sequences, how to model restriction sites. Now we'll start talking about alignments, where things will get even more interesting. If you're not so pumped up so far, this is very intuitive actually once you get it. But how we will arrive at it often will not be very, very trivial. It is one of those problems which once you see the solution, it looks like it's obvious, but it's very hard to get it the first time. So the premise of this is actually, this is one such contribution which came from mathematicians and probabilists directly contributing to biology. And this started in 50s, or actually even before 50s. Stan Ullm, he said, ask not what mathematicians can do for you, but what you can do for mathematics. He was addressing he was addressing biologists. They were all biologists in the conference. And this is his famous quote. He wrote a bunch of seminal papers which laid down the foundation of how sequences should be aligned. And we'll talk about why sequences should be aligned. Does anyone recognize this person? Like, who was? Yes, so he's involved in the Manhattan Project. A lot of innovations happened during the Second World War, and he was one of the contributors to it. But he also started this field of doing sequence alignment overall. He laid down the foundations of doing sequence comparison. In his paper, this is a review paper from another seminal person in this field called Waterman, Michael Waterman. He was, the reason I chose my grad school was because he is considered, again, depending on whom you ask, there are different fathers of computational biology, but he's definitely played a seminal role in computational biology. And one of my main reasons to choose the grad school that I chose was because he was there. However, often by the time I joined, he had chosen not to take students. So he had taken a soft retirement, though he used to hang around. And one of my most special feelings is, despite having a very terrible sort of grad school journey and defending during COVID, Waterman happened to come during my defense out of nowhere. He had, of course, he had other things to do. He's in his 80s now, or maybe 90s. He could be retired. He could have just chilled out at his home, but he chose he used to do that. Like, he would come for, he's still around, by the way, but he used to come for every defense just to, I think, keep learning, but also to tell people that he's still around. And, it's a very interesting feeling because the fact that I saw him during my defense itself changed how I just felt very happy. So it just changed how I was going to defend. Okay, with that short story, we'll switch on to the actual problem. The problem is of a lot of computational biology often is understood by sequence searching. We have found an organism where you're trying to figure out the function of a particular sequence, right? So you happen to come across a new organism, you somehow happen to know what gene sequence it has because that technology now exists. You would like to understand what is the function that this particular gene does. Now, these experiments where you have to actually figure out the function of a particular gene sequence is very, very hard. It could, for example, be involved in cholesterol biology. It could, for example, be involved in synthesizing hemoglobin. It could be doing anything on this earth, right? There is only one way to figure out, which is to actually do a knockout experiment where you knock out that particular gene and then ask that, okay, what happens to the organism? Now that is a hard experiment because it requires a lot more drudgery, to put it in simpler words, if you want to pull out that experiment. However, what is possible is during the course of evolution, different organisms have shared sequences. So we all, at some point evolved from one common cell. The fact that we evolved, we would have retained newer sequences, but the fact that we are all functioning and we are all living organisms does mean that we share a lot of sequences. So one of the facts that is invoked in biology is, if I know the function of a gene which has a particular sequence in one organism, can I try to see if the sequence that I have now found of a new organism, does it map to the sequence that I found? So for example, in human, you found a sequence which looked like this, and you know that this is doing, this is processing your hemoglobin, right, your HP. Now this is your new organism that you happen to sequence, and among different sequences, you still find AA, TT, AC, which is very similar to, what you had in human. Now if I map or align these two sequences, I can make a claim that, given that in this human, where I know the function of this gene, I can take a guess that it's very likely carrying out very similar function in the other gene as well. And broadly, that's the idea of alignment. For example, this is a famous example. There is an oncogene. Oncogenes are genes which are responsible for initiating cancer in one way or the other. There is this particular oncogene which is called, has a lot of similarities with PDGF. So this is an oncogene which is coming from a virus. This is called VSIS. It causes uncontrolled growth and it causes cancer in monkeys. However, it is also found to have sequence similarity with an actual human gene, which is called PDGF. The hypothesis was as follows, that given that viral sequence is required for inducing cancers or one kind of cancers in monkeys, is it possible that given this particular virus shares sequence with an actual human gene, we could also be getting cancers if I turn on or turn off this particular gene, right? Because the same sequence is conserved and this has a particular function in monkeys. In this case, we also share a particular sequence, which means that we will produce similar RNAs or we might not produce RNAs if I switch off this gene. So is it possible that if I try to find out the function of what this particular sequence is trying to do, and if I align it, can I deduce something from it? Can I deduce that this is also going to induce cancer in humans if I switch it off? Right. So in other words, is this the right gene which is doing the right thing but at the wrong time? Right. So if I switch off this particular gene, for example, it's a growth. It's a gene which is responsible for inducing growth. If I switch it off at the wrong time, I will end up getting cancer. Okay. So when you have two different sequences, there are only 5 or 6 possibilities that can happen. One is you can have a substitution. I'll show you the pictures and it will become clear what a substitution is. You can have an insertion. You can have a deletion. You can have a segmental duplication. You can have an inversion. You can have a transposable element insertion and translocation, like too many words here, right? But we will only focus on the first 3 words, which is substitution, insertion, and deletion, which is very, very easy to understand. So in the first case is your normal sequence where again this is your DNA, this is your RNA. So A is paired with T and so on. This is just showing you what each base corresponds to. If I have a substitution where I should have a GAA, a substitution leads to the G being changed to an A. Okay, that's called a point mutation or a substitution. You can also have an insertion where here I end up adding an extra base altogether. For example, in this case, this was CTT. Now this becomes CCT because I have added one extra base and everything else has shifted. So this is called an insertion. Similar way, you can actually remove this particular base from here and this becomes a deletion. In this case, I've removed an A which existed here and now I have a deletion. We will focus on the left part of the story where when we try to align 2 different DNA sequences, we will assume that they will either have a substitution or they will have an insertion or they will have a deletion or they will map perfectly normally. There is another paradigm in which changes can happen, and for example, there could be a duplication, which means that this particular gene during the course of either within the same human or across organisms, it could end up duplicating itself. The difference between an insertion and deletion is a duplication is a larger event. You can think of this as an insertion of the entire gene altogether in this particular case. You could also have a deletion, which means that I could end up entirely deleting this entire particular segment of the DNA. There could be an inversion, which means that what was supposed to be left and right now becomes right and left, and vice versa. You could also have a translocation from a second chromosome, which means that one part of this particular element is now relocated to the other either other chromosome or other arm of the chromosome. For this particular part, and most of the course, we will focus only on these events. However, a lot of diseases happen because of this also, right? So there are two premises of studying what we are going to study. One is being able to annotate the function of a gene which is not known, where you will try to align it to something where you know the function. The second is to understand, are there DNA mutations that have happened in my own DNA? Okay. So for example, if I have access to my sequence and if I align it, how do I align it to your DNA? In what ways should I take my sequence and try to align it? We are going to see the methods part of it. So some terminology before we do this. We will define this term called alignment, which is where we write one sequence on top of the other. So we'll just take a very simple sequence data. This is an alignment. I write two sequences on top of each other. There are certain notations. In this case, I will try to align, right? Why is it called an alignment? Because bases are matching. This is a match. This is a mismatch because I can align it, but it doesn't really match here. I could also do insertion. So I could say that there is an insertion here. There is an insertion in the second sequence, or there is an insertion, there is a deletion in the second sequence, or there is a deletion here, which is equivalent to having an insertion in the second sequence. So insertions and deletions are contextual. In this case, I will call it a deletion because this is in this sequence, I deleted this base. I could also call that this is an insertion because I ended up inserting this in this space. So that particular part is contextual, but hopefully that is clear. A dash represents an insertion or a deletion. The important fact that we will take into account is often you will not have a dash aligning to a dash. That has no meaning. It mutually cancels off. Okay. Once dashes have been inserted, sequence must have the same length. So in this case, the length of each of them was 4 to begin with, but I could have also started with a sequence only of length 3, but after I have added the dashes, it becomes a length of 4. Okay? That's the terminology. This is called an alignment. Okay? Is that terminology clear? Okay. So for example, I have these two sequences, ACG, TCTAG, and ACT, CTAG. Okay, let me write this down. And let's talk about how will we do the alignment. The goal of alignment is so that I can recover most of the bases to be same. Okay, how do I align this? So in this case, I've gotten 2 matches, 5 mismatches, and 1 not aligned. Can you do something better? Can you try to align more number of bases? Okay. So I will keep this as the constant, and what should I write here? How should I align? Okay, so I'll align T, C, T, A, G. Okay, and then add a dash. Okay. Anyone, any other alignments possible? I can delete G. In where? This one I should delete G. So deletion and insertion are like they're tied to each other. If I delete here, that means an insertion here. By deletion, you mean like so I cannot at any point I cannot reduce the length of the sequence. I'm not allowed to do that, right? So if you think that I can gain more by just deleting it so that everything aligns, that is not allowed. Okay, the sequence cannot change. The sequence has to be the same. You can add or insert bases, but you cannot actually simply delete things without it coming anywhere else. So I could do this. For example, this is valid. I could align, I could have inserted G and okay, this is valid, for example, right? I could align it, I could do a mismatch, I could align it to a particular base, but I can never delete this. Okay. Any other alignments possible? Yeah. I can do insertions and deletions can happen at any point, right? Because at this point you're working with the DNA sequence. So these, it's possible that during the course of evolution, there's no context to it, right? I think your question was your question related to, no, there are no codons here. Was your question related to codons? Hmm. Mm-hmm. Yeah. Yeah. Okay, so you're saying one more alignment, right? You're saying if I do this, I have lesser bases in the first sequence, so there has to be an insertion at some rate. Is this what you're saying? Yeah, you could do this. Which one is best? Which one would you pick? Okay, why? Yeah, so you're trying to maximize something, right? You could come up with different notations. You could actually come up with this particular version as well, where you have 5 mismatches, 2 mismatches, 1 is not aligned. I could also do 7 matches, which is what, for example, all of you invented, where 1 is not aligned, right? But you will notice that all this choice has been arbitrary so far, right? When should I align? When should I align? How should I match? This needs a scoring system. You need to be able to score. For example, if you have ever done optimization on machine learning, you try to optimize a score. In this case, there has to be a scoring mechanism so that you can actually do a proper optimization of that score. And that's what we're going to see very quickly today. Okay, that's a combinatoric problem and a fun problem. So let's talk about, given these two sequences, how many number of ways can I align this? This is a combinatorics problem, right? So it's a counting problem. If that term is new or if that term scares you, it's just a counting problem. How many number of alignments are possible? 7. Okay. Anyone else? How many number of ways can I align two different sequences such that I can allow for a match or a mismatch or an insert or deletion? Okay. A match means A to A. A mismatch means, let's say, A to T or G to C or A to C and all possible combinations. And an insertion deletion means A is just example there. It could be anything. 8. How? Why? Why 8? Okay. Okay, let's see. Anyone, any other answers? 8.
It's larger because you can have like you can just do multiple insertions deletions show that you are matching, but it's not the most optimal match.
So we are never allowed to match this and this.
Yeah, we
Right.
can have like basically
Hmm.
I have like in the second.
So his answer, yeah, so good. So his answer, where is it? So his Madhu's answer was that I can keep cycling the one insertion. Okay. So what is missing here?
Like, let's say we are looking at the second letter in both sequences.
Okay, this one.
Yeah, in both of them, like in the first one and the second one.
Okay.
A and C. So we can just do A and the, like we can increase the sequence length by 1, and then we have like an insertion on one side and a deletion on the other.
Okay. So let's see. That's very good, right? So it has to be larger. It has to be a function of the length, right? There is some. If I started from, for example, if I started from this sequence, how many possibilities do you see? The length is now shortened. There are only 4 bases. Okay, it has to be some exponential, right? We'll see what it is, right? So one way to think of an alignment problem is to solve a grid problem. So if I lay down my sequence A1 to AN, and if I want to align it to, let's say, B1 to Bn, you can think of this as a walk along the grid. Okay. We will develop some terminology to understand why is it a walk along the grid. But imagine if you start from, your goal is to start from the start, where you have either access to an insertion in both cases, and then you have to end up towards the last sequence such that you will end up aligning both of them, right? This is Bm comma n, right? There are m bases in the first one and second one has n. Now each walk that I do, I have 2 choices. I have 3 choices. I can either go this way. When I go this way, I'm moving along this sequence, which means I'm inserting deletions in the B sequence, right? So when I go, for example, this way, I'm inserting one particular, let me just make this clearer. If I go from A1 to A2, directly, horizontally, I have not moved from B1, right? My B1 is constant. Imagine your A1 was aligned to B1. What I'm saying is this has to be a dash. Okay. There is a second possibility, which is that I go down this route. So if I go down, I go from B1 to, let's say, B2, in which case I'm inserting an insertion upstream. And then there is a third choice where I go along the diagonal, where I'm saying that now A2 will align to B2. This length is M. This length is N. Can you think now? How many number of possibilities exist? At every point, I have 3 choices. I can either go left, I can either go down, or I can go along the diagonal. Mismatch or mismatch, deletion in A sequence. This one is deletion in B sequence. Does that make sense intuitively? Let's just take a very, I'll just end. Just take a very simple example of aligning what to y. So if I just write this down, what do I do here? Assuming the first start is a insertion in both, I don't want to start with an insertion. Even if I start with an insertion, I'm going to make a move this way, right? I will align W to W. What do I do next? I'm here now. Should I take it down? Should I take this? Should I take diagonal as well? Okay, what next? Okay, yeah, I could either go this way or I could go this way. And the moment I land up here, I have to end. So depending on how and where I land up, I will either go this way or I will go this way. How many number of possibilities? Let me, erase this. Yeah. So at this point, I'm choosing only the optimal paths, right? But I could have chosen anything. Sorry, go ahead now. Okay. So I can either Mm-hmm.
After we can have a deletion of T as well.
Sure. So you're saying but this, when we chose this was an optimal path, but it did not have to be. Like I could have indeed gone in this direction or this direction, right? So this is just to give you an intuition that it's going to be exponential. It has to be exponential by definition. So just think about, given a grid of m cross n, where you're allowed to choose only two paths, 0, 1, which means that you move this way. So this is this way, or you move, this is xy, this is this, 1, 0, you move this, or 1, 1. How many possibilities exist such that you land up from 0, 0 to nm? And we'll continue from there on Thursday. Please also bring your laptops. We will try to see how to align two different sequences using programs that already exist, and internally, how do they work, right? It's an optimization problem. It's a combinatorial, exponentially large problem. How do you solve that problem is what we're going to see on Thursday. Okay, thank you.